The next organelle we'll discuss is the Golgi apparatus. So this looks very similar to the endoplasmic reticulum in that it has many folds in it. The structure consists of a cis face, a trans face, and the cisternae. So the cis face, it's kind of like the receiving end, and this is where the transport vesicles come from the endoplasmic reticulum, as you can see from this picture right here. Then the material fuses with the cis face. It goes through the Golgi apparatus channels, and then it kind of discharges, and you can see that it kind of clips out, and they're discharged in secretory vesicles. So the cisternae part, that's where the modified glycoproteins and the modified glycolipids are going, and they're kind of like folded, stacked membranes. So these modifications, they might involve adding a protein to a sugar to make a glycoprotein, or they may cleave a sugar from a lipid or a protein. And the next slide, we're going to take a better look at the vesicles that help discharge the materials from the Golgi apparatus. So these vesicles also, like all the other organelles, they're membrane bound, but they store enzymes. There's two main types. We have lysosomes and microbodies. So the lysosomes, those are connected with the Golgi apparatus. They help transport the items, and they also contain the degrading enzymes, which help break down the proteins, the lipids, carbohydrates, and some nucleic acids. And then we also have the microbodies. They're unlike liposomes in that they grow by incorporating the lipids and the proteins, and then they divide. There's two examples, the glycosome and the peroxisome. Glycosomes have enzymes which convert fats to carbohydrates, and peroxisomes are oxidative. They have enzymes that remove the... The next organelle we'll discuss is the mitochondria. So mitochondria are known as the powerhouse of the cell, and this is because oxidative metabolism occurs here. It allows the micromolecules to store ATP. So the structure, you can see it's a smooth outer, smooth membrane running throughout the structure. It has many folds and layers known as cristae. So the inner compartment and the inner membrane space is on the outside. The main function of the mitochondria, as we said, is to facilitate oxidative metabolism and give energy to the cell. But mitochondria also have their own DNA. And what this does is it contains genes. It helps make proteins that are needed to facilitate the oxidative metabolism. However, this DNA, because mitochondria has DNA, this doesn't mean it can exist on its own. It needs to be within a cell. Next, let's look at the chloroplasts. And if you take a look at the picture here, you can see it looks very similar to the mitochondria. It has similar structure, but it's a lot more complex in that it has grana, and these are enclosed compartments of stacked membranes. And there's also thylakoids, which are in the grana, and these are disc-shaped structures. So surrounding, there's a stroma, and this is the fluid matrix that surrounds the cells. So similar to the mitochondria, the chloroplasts also contain their own DNA, and this DNA is used to make proteins that are needed for photosynthesis. And again, chloroplasts, you'll typically only see this in the plant cells. You won't typically see it in animals. So plant cells, a main difference between plant cells and animal cells are the vacuoles they contain. Vacuoles are really large compartments, and they're usually found at the center of the cell. So this is kind of like the vacuum of the cell. It increases the volume of the ratio, and it applies pressure to the cell membrane. Another main difference between plant and animal cells is the cell wall. It's not found in animal cells. And similar to the prokaryotes, the cell wall in the plant cell, it's used for protection and support, but differing from the bacteria cells, it's a lot more complex. So there's a primary wall, and this is laid down when the cell's still growing. There's a middle lamella, and this glues the cells together. And then the secondary walls, these are placed inside the primary walls. All right, that's the end of the lecture. Thank you for visiting myotherprofessor.com. And please refer to our website for more lectures. And also feel free to leave some feedback if you have any suggestions or other information you'd like to see covered. Thank you.